Alrighty then. On this lecture we're going to look at mechanisms of regulating the GFR, the glomerular filtration rate. So let's start with a bit of a review. Remember that the glomerular filtration rate is approximately 180 liters a day. And that remains relatively constant despite changes in the systemic blood pressure as occurs when you exercise or when you hang out on the couch. Blood pressure goes up or down. So when it goes up, the systemic pressure goes up, but the glomerular filtration rate remains the same. So we're going to look at ways by which that is accomplished. So something to remember is that the hydrostatic pressure in the glomerulus remains at about 50, and that's how we're able to maintain the relatively stable GFR. If that hydrostatic pressure, and this probably should read hydrostatic pressure, maybe I'll put it in the G for glomerulus, just so that we're clear on what this hydrostatic pressure is. That's the pressure in the glomerulus, in the glomerular capillaries. And we showed that was about 50 in a previous slide. So that has to remain at about 50 at rest and during exercise, so that the amount of filtrate pushed out of the glomerulus into the Bowman's capsule doesn't increase significantly. So there are a couple of ways that we're going to look at that this is accomplished. There are autoregulatory processes. And the autoregulatory controls, and these are intrinsic, which means that they are contained within the kidney themselves. So these two mechanisms, these two control processes, do not require any outside input. No hormones from the adrenal cortex, no sympathetic neurons. It's all intrinsic within the kidney itself. So we're going to look at the myogenic response, smooth muscle contracting once it's stretched, and then we're going to look at the tubuloglomerular feedback mechanism between the distal convoluted tubule and the glomerulus that I alluded to earlier. After that, we'll look at a couple of extrinsic controls that regulate GFR. So extrinsic, these are controls that originate outside the kidney. We'll look at the role of the sympathetic nervous system, and we'll look at our old friend, the hormone, atrial natriuretic peptide, or ANP. So we'll start with the myogenic response. And the idea with the myogenic response, if I can draw a quick glomerulus here, doesn't need to be fancy. I guess it should look like a glomerulus though. So afferent arterial. And the idea is that we're trying to maintain constant GFR despite increases in systemic blood pressure. So let's say that we have an increase in the systemic blood pressure. Gotten up, we're exercising, we're running around the block or something. And what happens is the blood pressure goes up and the arterial then stretches. So with increased blood pressure, that's going to lead to increased stretch. Of the afferent arterial. Makes sense. Pressure goes up, blood vessels will stretch. Afferent arterial. So, I'll try to represent a stretch with a dotted line. So the diameter is increased. We started with a small diameter, and now we've increased it rather dramatically in this drawing. But that's the idea. And that leads to increased flow, increased blood flow, and that, of course, will lead to increased GFR. And that's what we're trying to avoid. So what happens is that the smooth muscle reflexively contracts. You take smooth muscle and you stretch it, and it just basically constricts as a reflex. And that smooth muscle then, if we can quickly draw another glomerulus. Oops, that's really bad. Let's start over here again. Put my frowny face back in. Something like that. And remember that smooth muscle is wrapped around the afferent arteriole. So these are those granular cells, those JG cells, which are modified smooth muscle cells. They constrict. They were stretched and then they contract. Smooth muscle cells. Again, these are, they go by a variety of names. JG cells for juxtaglomerular cells, or granular cells, as some books call them. Okay? So they contract. And that leads to, say, increased resistance, which is going to lead to a decrease in blood flow, which is what we're after. We don't want the increased blood flow from the increased blood pressure. And after the increased blood flow, or decreased blood flow, that's going to, of course, decrease the GFR. That was our whole goal. So, smiley face, back to homeostasis. Okay, pretty simple. Myogenic response. It's generated right in the smooth muscle itself. Increased blood pressure, increased stretch. Smooth muscle contracts by reflex, reduces the blood flow, reduces the GFR. That's what we were after. So that one's pretty straightforward. The tubuloglomerular feedback mechanism isn't much worse. I made a typo in this, so first thing I'm going to do is block out this. Didn't mean for that to, that was a copy and paste error. So we'll just get rid of it. There. Okay. So, again, we're trying to regulate the GFR. And we're trying to slow it down when the systemic blood pressure goes up. So this tubuloglomerular feedback system, it's between the DCT and certain cells in the DCT, which are the macula densa cells, I'll point out, and between the JG cells, those juxtaglomerular cells. So on this illustration, the macula densa cells are written kind of small in the print. Maybe I should have put a box in there and enlarged it. But these are the macula densa cells. So the DCT, remember this is what I was getting at earlier, is this piece. In earlier slides, I tried to show the distal convoluted tubule right at the end of the ascending limb, the loop of Henle, which is what this figure is showing. But it's at the end of the ascending limb or the beginning of the DCT, same thing. It goes between the afferent arteriole and the efferent. So here is the afferent arteriole and the efferent arteriole. And it's this combination here. The macula densa cells are these cells that are shown in gray. They're part of the DCT. They're specialized cells. And so the way this works is if we have increased GFR, so at this point we have excess fluid. So we have an increased GFR. So excess fluid forced out of the capillary, flowing through the nephron. It flows through the PCT, it flows through the loop of Henle, and then it comes over and it goes up the loop. So maybe we could just sort of draw something simple to remind us of the flow. It's going to flow through the PCT, and then that's going to flow through the loop of Henle. And then that's going to flow through the distal convoluted tubule. And we will have an increased flow rate coming out of here. That's supposed to be increased flow. Increased flow past the macula densa cells. So let's go add a few bullets over here and say if we have an increase in GFR, then, I like these if-then statements, if increased GFR, then we have increased flow past the macula densa cells. They're in the DCT. And the macula densa cells are able to sense the increased flow. It has something to do with the ability to transport sodium across the membrane. And with increased flow, that transport is affected. That's not the important point of the story. The important point, if you remember, is that those macula densa cells are able to sense the increased flow, and the macula densa cells then release paracrines. Remember what we're trying to do? We're trying to slow down the GFR. So the macula densa cells release some signal molecules, local acting hormones that we'll call paracrines. And the paracrines then target the JG cells. And what do you think the paracrines are going to do, to the JG cells? Let me change color here so I don't doesn't get confusing when I run over the green here. So here come the paracrines, and they target the JG cells. Well, what would be a reasonable response? We have increased flow, we're trying to cut that down, so squeeze the afferent arteriole, slow down the blood flow, slow down the GFR. So these paracrines cause increased vasoconstriction of those smooth muscles, causing the afferent arteriole to constrict. Vasoconstriction of the afferent arteriole and that leads to a decrease in the GFR. That's what we were trying to do. So everything's happy at that point. So that's the tubuloglomerular feedback system. The tubule being the distal convoluted tubule and the glomerulus capillary of the renal corpuscle. The story again was with increased GFR, there's increased flow in turn past the DCT, which is detected by the macula densa cells, which release paracrines that clamp off the afferent arteriole and restrict blood flow, thereby reducing GFR. So those are the two intrinsic pathways. And the next slide summarizes this slide a little more formally and a little more neatly. So it's still a picture of the tubuloglomerular feedback. Helps with autoregulation of the GFR. So the figure's kind of nice. You just follow it by the numbers. Step 1, GFR increases, and that's over here. So excess fluid is flowing through the PCT, etc., through the loop. So step 2 was flow through tubule increases. Step 3, flow past macula densa cells increase. That's what we were just talking about. And then step 4, paracrines from the macula densa cells to the afferent arteriole. So the macula densa cells are the blue cells, 4. They release paracrines. There's a little short little arrow going to the afferent arteriole, which causes the afferent arteriole to constrict. Resistance increases. Hydrostatic pressure in the glomerulus goes down. That all makes sense. And we have a drop in GFR. So, summarizes the other slide more formally. So those are the two intrinsic autoregulatory mechanisms for adjusting GFR when systemic blood pressure goes too high. So let's do the extrinsic controls for the regulation of GFR. We mentioned sympathetic nervous system, which we'll do on this slide, and the following slide is with atrial natriuretic peptide. So this is nervous system. So you remember here now, the problem is when the sympathetic nervous system gets activated, we're generally trying to raise blood pressure. Fight or flight, blood pressure goes up. So the previous two mechanisms were trying to bring the GFR down because the blood pressure was too high. So this is a different situation. So think about it for a minute. It's stress. And we have a loss of blood volume. That's stressful. Just thinking about that, how would you expect the body to respond? We have a drop in blood volume, and our story is in the kidneys. So do you want the kidneys to keep filtering fluid out of the blood and potentially sending it to the toilet? Or restrict kidney activity and don't filter as much? Yeah, the latter. So we'll start with this stress of decreased blood pressure. So let's start here. And what we want to do, of course, is now raise the blood pressure and retain fluid. So you remember that the principal way of detecting blood pressure is through these baroreceptors, the carotid and the aortic bodies. And then from the baroreceptors, there is a sensory pathway that goes back to the CNS. We'll just do it like this. And this is sensory. Have to communicate with the brain what's going on out there at the heart and the blood vessels. And then there are a variety of outputs. So let's see, what do I want to start with? So keep in mind that we're going to be trying to raise blood pressure. So a lot of this we've already looked at because we've already done the baroreceptor reflex. But let's take this one and go straight to the adrenal medulla. One neuron in this case, and coming out of the adrenal medulla, which hormones? We have epinephrine, which I'm going to abbreviate as E, and we have norepinephrine, or NE. Most of what comes out of the adrenal medulla is epinephrine, and these, of course, are traveling by the blood. They're in the blood. And they are going to cause the JG cells, the afferent arterioles, to vasoconstrict. We've already done this when we're talking about systemic vasoconstriction, and that would lead to increase in systemic blood pressure. So, don't forget the other things that we've already talked about as far as vasoconstriction at the systemic level to raise systemic blood pressure. Here we're focusing on the kidneys. So this would cause constriction of the afferent arteriole. And what would be the benefit there? What's going to happen to the GFR? Go up or down? Yeah, GFR is going to go down. So decreased GFR. Does that make sense in this context? It should, because we're trying to retain blood volume. We don't want to lose anymore. So close off the kidneys. So that's one thing that happens. And another thing that happens is, now we're back to our standard 2-neuron pathway for sympathetic nerves. We're going to come in here back to those JG cells again. And they're going to vasoconstrict, but they are also going to release renin. So coming out of that is renin. Remember, renin is all about RAS, and RAS is all about raising blood pressure. That's the R in RAS, is the renin. And the end result of RAS, you remember, is to make angiotensin II. And we had a whole host of targets for angiotensin II, all designed to raise the blood pressure. Well, one of the targets of angiotensin II was the adrenal cortex. To do what? Right, to release aldosterone. So aldosterone is released from the cortex. And you remember the target for aldosterone. It's what we're going through, or we will be soon. We've talked about this a little bit, but the target is the kidneys. Remember on a previous slide in a previous lecture, we talked about the target for aldosterone being the distal portion of the DCT in the cortical collecting duct. So what it does there is that it increases the sodium retention, and that's going to increase the blood volume. Water follows sodium, increases blood volume, and that, of course, is going to lead to an increase in BP, which is what we wanted. Because our stress starting out was a drop. So here we're going to raise blood pressure. And there were several additional targets for angiotensin II. See if you can review that. So let's just put up here other targets. Other targets, and they all are working to raise blood pressure. Okay? So the sympathetic nervous system involvement, detected by the baroreceptors, sympathetic response to raise blood pressure. One response is to release norepinephrine and epinephrine, and that stimulates GFR reduction by vasoconstricting the afferent arteriole, as does direct nerves going to the same muscle. Because remember, this is norepinephrine and epinephrine coming from the blood. N-E and E, from the blood. It's a hormone in this case. So that targets the JG cells, as do neurons that directly connect to the cells, and they cause contraction of the smooth muscle as well. In addition, they cause the release of renin, which starts the RAS cascade for the production of angiotensin II. Angiotensin II has a number of targets, one of which is the adrenal cortex, causing release of aldosterone, which stimulates the kidneys to retain more sodium, increases blood volume, increases blood pressure. Kind of neat. See if you can work your way through that story and try practicing it and drawing it on your own. The last slide in this lecture is looking at atrial natriuretic peptide. So, different story now, so keep them straight. Atrial natriuretic peptide, you remember, is a hormone that's released from the atria of the heart when they're overstretched. So in this story, we have an increased blood volume, not too little like the last example. Here we have an increase in blood volume, and that's leading to an increase in blood pressure. But atrial natriuretic peptide is released when the atria are stretched. So what I probably should have done before jumping to increase blood pressure, let's do it this way. Let me get rid of that. Let's say we have increased atrial stretch. Let's start with that. And that's what I've tried to show on the heart. Normal heart, solid line, dashed line is expanded from increased blood volume. So increased stretch, and what comes out of that is the hormone atrial natriuretic peptide. So I'll just write ANP. And this also goes into the blood. And then it has a number of targets. So keep in mind what we're trying to do. Blood volume is too high. So now we're trying to get rid of excess blood volume. Well, atrial natriuretic peptide targets the afferent arteriole. And what do you think it does there? Trying to get rid of blood volume. Yeah, so it's going to increase vasodilation. Increase vasodilation. And what's that going to do? That will cause increased blood flow and increased GFR. So in this example, we want to increase the GFR to get rid of the extra fluid. And we wind up with an increased GFR. And that is going to lead to a drop in blood volume, which is what we want. That was our stress that we started with. Here we have reduced the stress, so we sort of closed that feedback loop. And this will also have an effect on lowering blood pressure. But the story is about blood volume. Blood pressure is sort of indirectly related. And the other thing that atrial natriuretic peptide does at these JG cells is that it also inhibits the release of renin. inhibits release of renin. And that is going to lead to a decrease in RAS, which will lead to a decrease in blood pressure also. Okay? So atrial natriuretic peptide has a couple other targets. One other target here is the nephron. And remember when I first introduced the nephron in that busy figure, talked about the regulation of sodium at the DCT and the cortical collecting duct, and atrial natriuretic peptide caused the loss of it. So we retain less, basically. So we are going to have a decreased reabsorption of sodium. Decreased reabsorption of sodium. And that's going to lead to decreased fluid retention. Water is going to follow the sodium, and we're not reabsorbing it, so the sodium is staying in the filtrate. Water follows the sodium. Decreased fluid retention, and that leads to a decrease in blood volume. Okay? And atrial natriuretic peptide also targets the adrenal cortex. And what's important in the adrenal cortex for blood volume is aldosterone. So are we going to release more or less aldosterone? We're trying to reduce blood volume. Well, aldosterone is part of RAS, which is all about raising blood volume and raising blood pressure. So ANP is going to be working sort of antagonistically to aldosterone. So we will say we're going to have a decrease in aldosterone release. Remember that aldosterone promotes sodium retention. So that's going to lead to a decrease in blood volume. Decreased sodium retention, decreased water retention, decreased blood volume. Okay? Lots to practice there. Please ask questions, study it, concentrate on it, and let me know where you get stuck. And we'll start talking about reabsorption next. So this sort of wraps up our discussion of GFR and regulation of it. Increasing and in some cases decreasing GFR. Okay.